any animals who naturally have our disease. I don't know if there's any animal that naturally has CAG repeats. I'm going to say they're not. So we can't go and study a cat who has the disease because they don't ever have the disease naturally. I don't know about you guys, but when I got the call and said, we'd like to take pieces of your spinal cord so we can study your nerve cells.
You said no.
I said no. And the main reason is because I can't replace what they take out. And it causes really bad damage. So in other words, if you want to study the biochemistry of a guy with KD, they have to be dead. You can't do it. Have to go. So that means, how do you get around that? You get around that with different models. And so I'm going to go in and repeat some of what we said yesterday about kind of 3 of the major models that people might talk about in the next day or 2. And they are based, I have them in order of complexity, with the simplest and cheapest first. And the simplest and cheapest are just cells you grow in a dish. Now the bad news is they don't have muscles, they don't have neurons necessarily. They're not in a group like they are in your body, but they're really cheap and quick. And it's easy to do the experiments and do lots of experiments. And let me just show you sort of data you get from them. So this is a set of normal cells. And what they've done is they have the normal androgen receptor. They've added androgen. And so where should the androgen receptor be if there's testosterone around? In the nucleus. All those green circles are the nuclei of cells. All the androgen receptors, most of it, are sitting inside the nuclei just where they should. And so these would be normal cells. You're going to say, okay. Now what have you used with the same types of cells, one that had a very long CAG repeat? Well, you see some of the cells having in the nucleus, but all of a sudden you begin to see other things. You begin to see ones that have these clumps. So what are those clumps? They're the inclusions. They are the indication that you have too much of the particle. In other words, so now we have a way of seeing in the cells, even though we don't get degeneration of neurons or muscles or atrophy or any of that. But what you can see is whether or not they have the clumps, which is a signal they're not getting rid of the androgen receptor properly. And so you will see initial experiments all the time when they look and say, look, we got rid of the clumps by adding this drug. Woo! Now, the fact that it works in the cell, does this mean it's going to work in humans? And the answer is, of course not. Humans are much more complicated. So another animal model that's used, and this is really an amazing one, is fruit flies. What makes it amazing to me is fruit flies have no androgens naturally. They have no androgen receptors naturally. So they don't use testosterone. They don't even have it. But if you insert into them a gene with a human androgen receptor, remember because it's the same genetic code, they can make the protein. You can have them make the protein, and because of fruit flies, this is why they love fruit flies, you can have them make it in any cell you choose. So here they made it in eye cells. So if you can give them the human, and there's ways of injecting it, it's not an easy procedure, but it's pretty commonplace these days. You can give them the human gene into a fruit fly, and the fruit fly will start making a human protein. And in this case, specifically in the eyes. If you give them androgen receptors with short cues or long cues, and you leave the flies alone, nothing happens. Because remember, one of the things you have to get the disease is the testosterone. They don't make testosterone. So what do? You put it in their food. You put it in their food, all of a sudden you see in the ones with the long CAG repeats, you can see their eyes begin to fall apart and look funny. They have eye KD. I mean, this is remarkable to me, that an animal that doesn't even have the protein, it doesn't do any real function, at least theoretically, still can cause cells to die. And it's a nice model. There's reasons actually why this model is used, and it has to do with something we don't care. But again, it's not humans. So the next best thing to a human is a mouse. And although they won't admit it, there are about 4 or 5 different KD mouse models, and everybody who developed them is telling them how their mouse model is the best. It's kind of like Detroit and cars. And they all have different properties, and Some are good in certain respects and some aren't. But for the most part, I think most of the results have been pretty consistent. Now, how do you know if a mouse has KD? You can't tell you. Then this is kind of what we saw yesterday. There's a set of tests. The easiest, one easy one to do for any muscular problem is to pick it up by its tail. Pick it up with its tail and it's normal. Its feet go out like this. It squirms and of course eventually bites you. If it has a muscular problem and instead goes in like this, I don't know why, but it does. And so it's real easy to tell them they're muscle. Pick them up. If they're sprayed out, they're fine. If they're all crumpled up like this on the right, they have some sort of problem. So they would have KD if you gave them the KD gene. And again, it's possible to take the human gene, put it into the mouse. In fact, someone else has taken the mouse gene and actually put in a CAG repeat. So it's using the mouse gene that doesn't have a CAG repeat, puts it in, and you get the same symptoms as Kennedy. You have the walking test. A coordinated mouse walks with its back and front legs, it's perfect. And then it looks like how some of us were coming home last night. It's not that coordinated if you have the equivalence of KD. Your front and back feet are coordinated, doesn't look right. These are both qualitative tests. It's hard to say, Here's a quantitative one. This is one that's for what's called rearing. And mice, when they walk around, will go up on two legs, kind of like lemmings from that lemming show. And if you take those little laser things you see in the movies to detect when you're protecting jewels and stuff, if you put them at the top of the cane, you can actually have the machine count every time the mouse steps up. So you can put the mice in there and go home. Come back the next morning and have a whole experiment done for you. And what you find is that if you take a normal male mouse without having the Kennedy's disease gene or allele, you get rearing as these blue guys go. If you have a Kennedy's disease mouse, they're the red. And you can see they don't stand up as much. Now, mind you, it could be they're just not interested. But it's more likely the reason is because they're just not physically capable of standing up as well. That's the argument. To support that, what if you take female mice and you put in the Kennedy's disease androgen receptor gene? They don't have any testosterone. You shouldn't see any effects. And you don't. Their rearing is the same whether or not they are wild type or have the other gene, because they don't have testosterone. Of course, the rotorod, what could be more of a hoot than putting mice on a thing and watching them fall off? And you're tired. So what does the data look like? Here is the blue, the dark blue dots represent a wild type mouse, a mouse that's normal. Over time, and you can see it takes a while, they learn, but as they get older, they learn how to stay on the wheel for almost 2 minutes. And by the time, that wheel's going faster all the time, so it's pretty good. KD mouse to the pink, and they get down to where they have trouble staying on for more than 10 or 15 seconds. Again, because their coordination is messed up, their muscles aren't working. And so you'll see data like this. And of course, in humans, you do it by timing how long it takes them to fall off the rock. Now, so if we were to look for a cure for KD, these are the ways you can kind of looking, and cure is probably the wrong word, but a way of treatment. You can prevent the AR from entering the nucleus. So you can do that by, in mice, they castrated. That seemed to work. Or maybe drug equivalent that tries to lower the amount of testosterone. And that's what the leuprolin and to some extent the testosterone trials were trying to do. In a way that's not nearly as drastic and having the effects that a castration could have. And it could be that in humans castration doesn't work that well. Find a new way to remove the bad AR. And that's a lot of the drugs are trying to stimulate that. Is there a way we can get an alternate pathway? Find something new and spicy. This has to do with an ASCJ9, because it came from whatever cumin or whatever that is that it comes from. But essentially what that does, if I understand it correctly, is it deals and interferes with the androgen receptor's ability to interact with some of those co-factors, those co-activators. Decrease AR concentration. What if there's a way of just not making as much AR? And I think tomorrow you might be hearing something, a study about that. I might have misread it. Is there a way of somehow decreasing the amount of AR that's being produced? Or maybe there's a way of not worrying about the neurons and working directly on the muscles. Is there a way of keeping the muscles alive and somehow get reinnervated by some other neurons? So these are all different things you may or may not hear about tomorrow, but it's the basic ways that you try to design a treatment. You know, and looking at what's going on. Does that make sense?
And one question on number 5 there. Is there ways to contract and expand the muscles without, is there a different way to stimulate
No.
muscles?
Not unless you take a cattle prod and shock you. So there are, I mean you laugh, but there are ways that you can attach machines that cause your muscles to contract. I don't know how well they work, but I mean you couldn't do it that way theoretically.
On number 4, would that not decrease your strength?
Yeah, it would decrease. I think your real answer is probably yes, but the idea is, are you willing to take a little bit less androgen effects, but to maybe you don't have to lower it enough that has major sexual side effects, but it does have, for example, it does reduce the ability to have the disease. I mean, the answer is you don't know. And I'm just making this up, so it's...
In your list of 5 items there, is it number 1 when you're talking about prevention of testosterone?
It can be. So if you were to castrate, that would keep it from entering the nucleus.
So, is there or has there been any consideration about the quality of the testosterone?
Well, that's kind of what testosterone does. Because testosterone comes in 2 forms. Testosterone, which is what's synthesized usually from your intestines, and dihydrotestosterone that's usually made, I believe, in the cells that use it. And the dutasteride block that conversion. And dihydrotestosterone is a lot more potent, so it takes less of it to cause the effects. It binds essentially better to the AR. And so the dutasteride lowered, I believe, it lowered the levels of dihydrotestosterone. I just want to make sure when they measured it. And therefore, but didn't affect testosterone levels per se, just the dihydrotestosterone. And therefore it was hoped, and it kind of worked. It was close, maybe a little bit longer. It was enough that it apparently tried to slow, it seemed to slow down. And we have to say seemed because it wasn't statistically significant. But it was close. So it seemed to slow down the loss of muscle strength during the trial. Did I say that okay?
Yeah, I have sort of a passing question about the quality and the chemical composition of testosterone.
Well, testosterone is
source and its chemical makeup.
when you make it yourself. And it doesn't matter, everybody's testosterone is the same. Otherwise it wouldn't be testosterone. Now if you're talking about having some sort of testosterone analog, something that you're ingesting, that's a separate process entirely. And that you need to talk to your doctor and probably the researchers. That there was a study, 15 years ago with adding testosterone to KD patients, and it did not have any effect. And I don't remember if it had any bad effect.
Well, I thought that I read that they did have an effect, but when they stopped adding testosterone, the cravings reversed back to their pre-state.
And when you say effect, a good effect or a bad effect?
Negative effect.
Yeah, that could be, as I said. I know it didn't have a good effect. No, that could be.
Another question for you. You were able to take, when you're doing a mice model or a fly model, you're able to change the genes in that to make the repeat.
Right.
Can you take that in a mice model and uncase those out?
Can you repeat the question?
She wants to know, is it possible to go in, and you probably have been talking for people as well, removing the AR gene. The problem is, as far as I know, technically there's no way of doing that to an intact person. Second, then you wouldn't have any AR. And so it'd be the same, essentially, probably as castrating. In that sense. I mean, in other words, you would be losing...
No, what I was thinking was, can you go in and change the DNA sequence? Can you go in of any type of that to have an effect on those mice? What the different...
In other words, can you add another gene? Sure, if they know what gene to add. The advantage of the fruit flies is that there are ways of looking at other genes and see if they interact with the androgen receptor and can lessen the symptoms. And if you can find that gene, and then you can see, well, does that also work in humans? And so maybe there's a way of doing that through some sort of drug. At this point, we're far away from being able to know and being able to technically be able to change the genes in an adult person. In the future, that might be possible, but right now it's very difficult. It's been rarely done in specialized cases.
I have a follow-up question. Given what you've described here as the blueprint to the end process where this all happens, we're aware though that the process starts at different times in different individuals and it progresses at different rates in different individuals, and in fact there's some relapses or there's periods where there doesn't appear to be any activity in certain individuals. So given that the engine and the blueprint is consistent, what are the variables and the parameters that affect the starting,
right,
progress, and
the real answer is they don't know. Okay. There is, and this is from me, I've been from a paper from about 5 or 6 years ago, and I just liked it because it seemed to make much sense. And then we're looking at KD, but we're looking at women HD, Huntington, so it's the same sort of thing. They have the same thing. Generally, if you have and Huntington's a little bit stronger in this relationship, but a shorter repeat length that gives you Huntington's, you have a longer, essentially, incubation period before you get the disease. A longer repeat means you get it sooner. So that if you have 100 or so, which we don't see in KD, they get it when they're teenagers, instead of when they're in their 30s and 40s. Now, there was a study done in mice in an animal model for Huntington's. And what they noticed was that if you took individual cells at different times of their age, the number of repeats was not constant. Most of them would be very similar to what it was originally, but it varied as time went on, it varied more. Most of the time it went up. So in other words, an individual cell can actually increase their CAG repeat and not affect all the cells around them. And so what they said was, wait a minute. They went and saw the cause of this, they thought, was because of a DNA repair enzyme. And so the cell doesn't have to reproduce. The cell just sometimes comes around and says, fix the DNA there. And you kind of almost imagine that when you're looking at CAGs, the repair enzyme loses count and tends to put in more. And so they then created a mouse that didn't have that repair enzyme. And what they found was that the amount of variability decreased and the time of onset of the disease was much longer. And so they hypothesized that what might really happen is that 40 is not enough to really give you the disease. You might have to be, let's say, at 60 or 50. But over time, some cells randomly get up to 60 and then you have the disease and that kills a specific motor neuron. The closer you are to 60, the sooner that's going to occur. So that might be why a longer repeat occurs quicker. Is that true? I don't know, but it makes sense to me. And I don't know if anybody can, any of the real researchers can comment on that. But
well, they talk about the anticipation between generations.
that's an increase in that's slightly different because that has to do with your sperm cells and your egg cells. So you're changing what you're giving off to the offspring. This is what's happening to cells that aren't involved in reproduction.
We don't see,
But it might be the same sort of process.
yeah, we also don't see anticipation in TV. What?
You don't see what?
We do not see anticipation.
We can't hear the questions.
She said, he asked about, is this related to the fact that in a lot of CAG repeat diseases, the repeat in children tend to be higher than in the parents. And this is called anticipation, that seems to get worse as generations go on. And Aline said that generally we don't see that in KD, right? I mean, you do see it in other points.
Oh, I thought in the literature I did read that there is anticipation between generations.
Yeah, I'm not an expert. Aline's with me.
It's pretty stable, that even between siblings, their CAG repeats can be very similar.
between siblings, but.
And between next generations, very similar. So if we look at a mother and a son, we have very, very similar repeats.
Okay. Now, if I have time, what time do I end?
10 o'clock.
Okay, what I'm going to do is something, and I'm going to ask Carlos for your apology because I'm going to talk about your paper. I just want to show you a recent paper that, and there's a couple things I want to point out, and just show you the sort of data that came out and that you can actually understand most of it. You might not understand what they did, but you might be able to understand the data. And so, if only knew what we were doing. And the disease is IGF administration ameliorates, makes better, disease manifestations. In a mouse model, spinal mobile, and Carlo, and I mean, one thing that I think is really telling about the case, I mean, a lot of times when I read on the forum, people are very impatient, not a lot, I mean some are, and they also impatient with the researchers, that why don't they work together, why don't they do this? Well, there are at least 3 major labs involved in this research with Kurt's lab, Maria Panuto's lab, and Gen Saboy's lab. And there might even be a 4th lab, I'm not sure. So I mean, this is like some of the major researchers working together. So there's a lot in our disease where people do work together. And so just think to yourself, what they wanted to test was whether or not IGF made the mice better. And so how would you design an experiment? And you know, then you think about it, you take a normal mouse, you take one that had KD, and one that had KD, and you inject IGF into it. That seems, and then you measure the things we've already talked about. Now what makes this really good is, remember all the things we talked about, picking up the mouse and the runaround? They didn't use those! They used a whole other set. So, for example, one of the things, and if I make a mistake, they did is they measured. Remember, one of the problems you think you have when you have the long, you know, the KD version of the gene, of the protein, excuse me, is that you have too much of it because you can't get rid of it. So they measured. Okay. How much AR there is. Now, don't worry about how they measured it. Just trust me. That you can see this line right there is the amount of human AR they put into the mouse. A darker color means more androgen receptor, lighter color means less. And if they treat it with IGF, is that, and now we're going to have, Jameson said he'd help me picking these out. Which one is darker? No IGF, IGF.
I'm going to have to say the IGF
It's
is lighter.
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 Okay. So in other words, there's less AR. That suggests it's getting rid of it better than it was in the other one. And you're going to say, well, that's just one example. What if you did it a whole bunch of mice? Well, that's what they did here. They measured it. And essentially, this one here, these are the KD mice. These are the KD mice with IGF. And it's significantly reduced. So that's a good sign. Right? Good sign. What about if you looked at the cells? And they're looking at muscle cells. And here, now, remember how all the other times when we see for AR, they always glow pretty colors? There's the green and the reds. Well, here they use a slightly different way of doing it. And what happens is wherever there's AR, there's dark black stuff. Okay? And muscle cells have lots of nuclei, but they're very small. So, for example, this is one. Okay. Now over here, there's one there. Now, these 2 sets of cells probably have the same number of nuclei, but which ones have more of the darkly stained nuclei? Jameson, one on the left or the right?
I'm going to have to say the one on...
Who has more?
Oh. Does that say vehicle?
No, vehicle's fine.
Vehicle.
That's just a control. So in other words, once again, these are the ones on the left are the ones that have the disease, the ones on the right have IGF. They were able to clear out more of the protein, which is, again, what we think we want to have happen. 2 for 2. What about just the mice in general? Now there's two mice, Jameson, one on the left, one on the right. I'm just going to say, which one would you rather be?
Oh, definitely one of the left.
Because?
Bigger.
Bigger. That's the one that had IGF, but still has Kennedy's disease. The one on the right has no IGF with Kennedy's disease. If we were to take a whole bunch of mice and weigh them, what we find are what's on the left. Now it's a little bit weird because there's basically three things you have to look for. This is the weight, the black and the green are the weights of mice that are just normal. KD ones are the dark blue. In between is, what color is that, Jameson? He can answer for himself, no cheating.
Green. Bad at colors.
This green. And this is the IGF. Now, notice two things. One is, they're certainly better than disease, but they're not as good as the wild type. Part of the reason might be, the way they designed the experiment, they didn't give IGF from birth. They gave it the way it would be done in humans. They gave it after they already started having symptoms. And so once you start having symptoms, it may not be possible to go all the way back to where you were. But it is better to get, you certainly are, much better than you would be without it. Well, then ones we don't want to see. The one on the bottom talks about survival. Another way of looking at survival is the higher up, the more likely they lived. And you can see that the ones that had KD in mice, they tend to die much earlier, but if they have the IGF, they don't die quite as quickly. And then up here is, so for strength tests, we have all the ones we talked about in mice, right? The run and the walk. Do they use that? No, they use a hang test. They take the poor mouse and hang it on a rod, is that right? And then they time how long it takes for it to fall. I know, it's so mean. But what you see is that if they have the, IGF-1, they do better than they do if they didn't have the IGF. So they maintain their strength better. Okay, this is the tough one. This is the one. Now, so far you've only been okay. One of those, the cells are smaller than in the other two. Is it number 1, number 2, or number 3?
I have to say number 2.
Everybody else?
Yep.
Yeah.
That's the KD ones. This is the wild type. This is with IGF. So then in fact it looks like the muscles stayed bigger.
Yeah.
And of course you're going to say, well, they just picked the data. Well, then they measured a whole bunch of mice, and what they found was the gray was KD, the black is the cross-sectional area of the muscle cells when they have KD and IGF. And they're almost back to normal.
Wow.
I'm already feeling buff.
And IGF-1 is FDA approved.
And they said yes. The bad news is,
Oh, no oral.
and I wasn't going to show this, because I'm not sure how they're going to interpret it, but I'm going to give you my spin, and then they can tell me why I'm wrong. This is an indication of counting the number of motor neurons in the spinal cord. Remember, they didn't inject until after they began to see symptoms. And what they found was there really was no significant difference, even though there was an increase in the IGF version. So the IGF had a few more, but not enough that you're not sure that it isn't just from randomness. And one could argue, and I don't know the correct argument, that, well, this is just an indication that it's going in the right direction and some reason we just didn't do it right. But it also could be, and I don't know if there's a way around this in the experiment, but IGF is a large protein. And you may not know it, but your brain is protected from things in your blood by this thing called the blood-brain barrier. It's like a movie. And I don't know if IGF can cross the blood-brain barrier. My guess is it can't. Very little. So in other words, you may not expect to see anything in the brain because the IGF doesn't get there. And yet we still saw the effect, so we must be affecting, well, either the muscles directly, which you probably are, and possibly maybe somehow affecting the axons that are coming in. But, so this isn't really a surprise. I'm going to leave here just to say that people, when you read the forums, as I said, get very impatient. And they're always talking about things like stem cells. And you always read about stem cell breakthroughs. And it reminds me of that quote from Apollo 13, where stem cells are the 1,000 things that have to happen. We're on number 8 when they have that breakthrough, and we have to get, 692 to even come close. So a lot of the things that we think are going to happen probably will, but not really soon. There's a lot of difficulties. And I'm tired.
Thank you.
All right, I want to thank Ed. We are going to be hearing a lot of technical jargon in the coming days, and now we have something to latch on to with the stuff that he explained to us. We have a much better understanding. I'm a science teacher. I teach high school science, and I tell my students, good science, we go to answer a question, and more often than not, that the answer to that question brings up 10 more questions. And that's where we are with KD research right now. We're making progress, but each question answered just brings up a whole other series of questions. 2 announcements. 1, tomorrow, Mike has negotiated with the hotel. We do not have to check out until 1 p.m. So you do not have to check out and carry all your bags to the conference center. We are able to meet here, and then we'll finish at noon, and you'll be able to go back to your hotel room, pack up, and check out then. And second is, Lou is going to share some things with us.
Okay, before everybody takes off for your break, I just wanted to